since 2018. It started with Dr. Sharan Ramaswamy and I back in 2018 over the Engineering Center, and it has grown into this. And this year, as Dr. Ramaswamy is enjoying a well-deserved sabbatical, Dr. Dickerson has joined the team and has helped organize this as well with some fantastic contributions from CellMet. So thank you all for being here. I want to thank a few people up front, and don't want to wait till the end to do this. I want to thank Ms. Claudia, Ms. Miriam, and Ms. Ingrid, who have done fantastic jobs getting everything together. They are outstanding. None of this could have happened without them. Claudia in organizing everything, Miriam in making sure the arrangements are nice, Ingrid in making sure all of our digital booklets are just absolutely perfect. So thank you all so much. Having said that, if you haven't seen it, there's a digital booklet out front. You can scan the QR code and you can see the agenda for today and all the happenings, including participant information and the One Blood truck as well, and the information is there if you want to give blood today. I would like to thank the Department of Biomedical Engineering. This is sponsored in part by the Coulter Foundation's endowment to the Department of Biomedical Engineering from CellMet, and also the Florida Heart Research Foundation, who has supported this event and has been great supporters of us, including approximately $1 million in funding to FIU this year that we're using to set up, we're using that to set up a whole new heart and vascular physiology core and a center here at FIU that's going to be outstanding. It's really going to take our cardiac and vascular research to a whole different level, so thank you for that. Having said that, first I would like to invite the Dean of the College of Engineering, Dr. John Balakas, to come up here and start us off this morning.
Good morning. Good morning. Welcome for those of you who are not from Miami and you haven't studied here, you're not studying here. Welcome to our campus, and enjoy the campus and enjoy the facilities that we have, and certainly take the time to enjoy this beautiful campus. We are the #4 largest university in the country, and we continue, our College of Engineering actually continues to grow. We've had 53% more students in the last 5 years. Our research has nearly tripled in the last 7 years. Our faculty are doing something like $320,000 expenditures per year. You know, that's among the top 10 universities. So we have incredible people, and you students are being taught by amazing faculty. I wanted to say that this is a wonderful event. There's nothing more than trying to help people, particularly, you know, when it comes to, you know, heart. So, and I want to say this is our 5th annual Miami Heart Day Symposium. Thank you to Joshua, Professor Hutchison, and thank you to Darryl. So today, Dr. Dickerson, good morning to you both, and thank you for organizing this and for all your time and energy and effort. It's really giving back. No one individually was tasked to do this, you know, symposium, but they do it. They do it because they love, because they want to serve, and I hope the students can see this example of giving back and feeling part of the community, you know, while you're doing it. And I want to thank our keynote speaker today, Dr. Chen. Good morning, sir. And also the CellMet director, who is here today, Professor Bishop. Welcome, sir, welcome. And his team. We really appreciate this collaboration among universities, in particular from, in this case, from Boston University. And it's the focus, heart, right? And then regenerative medicine at the same time, tissue engineering at the same time. I used to be that my research was about 50% biomedical, and I've written a couple proposals on tissue engineering. I didn't win them, but you can see how much my love is on tissue engineering. At least I can speak tissue engineering quite well, you know. In any case, thanks to also to Professor Agrawal, who is here, the chair of the Mechanical Materials Engineering Department, and Dr. Tremante. who are providing and leading from FIU the CellMed activity. And also, the executive director of Ms. Nancy Cavalli, for supporting our research and programs at FIU. And I wanted to say that this support of our research from NIH is amazing. Without it, we couldn't be who we are. We could not enable and contribute to the community, which so badly needs us. And it's a responsibility. We have a responsibility. It's not a job when you work in this area. So I want to transfer that idea. It's a responsibility. It's a responsibility that we have to the community. So I hope that the students feel that you are being educated to take a leading role and that you will play that important role in servicing our community for years and decades to come. Is it important to study hard? Let me tell you, it is very, very important. I would say every 40 seconds we lose someone because of a heart issue. Every 40 seconds. That is unfortunate. That is very unfortunate. So we need to think about these statistics. They may not happen next to you, but statistics tells us something. $229 billion are spent, or it's costing us in a variety of ways. Maybe loss of work, maybe hospital work, hospital costs, and so on. But it is very costly to our country, and therefore we want to definitely take care of that. We want to definitely help people live productive lives and not, having to deal with these things, with these issues, but rather make it a routine issue. Something can be detected, and we're developing sensors. I'm one of those guys who is developing sensors to detect, these things, early on. All right, I want to say that we're very proud here at the college to see our faculty and researchers also being, in this, working in this area. We've received in this related area of heart research about $5 million of grant just recently on this topic per year. So we are working on it. We are working on it. There are a lot of opportunities for our students, so please reach out, to your faculty to do more work in this area. We are, I want to close and say that Dr. Hutchinson is also conducting cardiovascular research on chronic kidney diseases, associated with chronic kidney diseases. And he is also one of our 8 winners of NIH R01 grants. And that's active at this particular moment. So plenty of funding for you students to work on and to make a difference. I want to close last week I was in DC, or 2 weeks ago I was in DC with other engineering and computer science deans. And I can tell you that the #1 priority for our nation is to create knowledgeable technology people, not only in medicine, but also in DOD, in Department of Defense, Department of Energy, security, cybersecurity. The need for people in AI, in cyber, in medical, in robotics, automation, the need for engineering, the need of fintech, the need of all areas in technology is so large. So please, tell your friends to come to engineering. Tell your friends to come to sciences. Tell your friends that this is the future of our nation, the future, workforce that is going to make our nation to continue to be at its best, to continue to be at its best. So you are our best. So please invite everybody else to join, this effort of creating, the wealth and also technology and also knowledge for our nation. So I want to wish you a very good day today, a interactive day, and a day that Dr. Chen will also start, here today, and will motivate. So we're all here. Please, you students, make yourselves available. Ask questions. Don't be scared. No question is small. No question is silly. It's really a way of interacting and talking with each other.
Thank you very much, Dean Velazquez, and good morning, everyone.
Good morning.
All right, good morning, everyone. Good morning. All right, it's good to see you all on this wonderful Friday morning for Heart Day. So I'm really happy to be here with you today. I think being a part of this event, we're here as researchers, as practitioners, as students, as educators, as members of the community to have conversations, as the Dean mentioned, have conversations about how we can take our work and bring it together so that we can have an impact in our communities for heart health. And it really gives me pleasure to introduce our speaker today, Dr Chen, who's done that through his work. And it's a through line of his work, if you've looked at his research and the impact that it's had over the years. So to share a little bit about Chris and his current appointments, he is the William Fairfield Warren Distinguished Professor of Biomedical Engineering at Boston University, the founding director of the Biological Design Center, and a member of the Wyss Institutes for Biologically Inspired Engineering at Harvard University. He serves as the deputy director of the National Science Foundation Engineering Research Center called CELMET, and a co-PI of the National Science Foundation Science and Technology Center for Engineering Mechanobiology. So Chris does a lot of stuff. He does a lot of stuff really, really well. And I think it's in part because in his work, he looks at the fundamental understanding of how cells interact with their environment, takes that fundamental understanding and looks at how to apply that, and then looks at how to translate it to human health. And I think you'll get a sense of that today in his talk. So I want to welcome Chris to the stage for his talk. Thank you very much.
I'm so excited to be here. It's my first time on campus, and I was just amazed at how beautiful it is down here compared to our gray, cold Boston area. Okay, so I think I just need to wait for my slides to come up. What I'm going to do today is talk to you a little bit about some of the work that we're doing in the cardiovascular space. But I want to start by just describing how important this problem is of heart disease. So we all know that it's the leading cause of death. We all know that it's something that's very common. I'm sure every one of you knows somebody who's suffering from heart disease or has passed on because of that. The cost to the medical system is gigantic. Part of that is because once somebody has heart failure, the kind of support that one needs to be able to stay alive, to be able to function, requires a lot of interaction with the hospital system. And one of the things that I didn't appreciate until I started working in this space is that many drugs, probably about 1/3 of all drugs that have ever been developed, have cardiovascular side effects. And some of those cardiovascular side effects are tolerable. For example, in the case of a life-threatening cancer and one takes the chemotherapy. But even in those settings, they are causing heart disease. We just accept that in the case of those hard choices. But there are many, many pharmaceuticals that are taken not for immediate, urgent, life-threatening diseases. And it turns out that many of them are discovered to have heart problems after they're on the market. So if you think about that, we test these compounds in animals. They don't seem to have any toxicities. We go into clinical trials. We test them in 1,000 patients. Doesn't seem to have any issues. And then the compound goes out and gets used. And often in that phase, some drugs are found to have serious cardiovascular side effects and get taken off the market because of that. And so you might ask yourself, why? How is it that we miss these major cardiovascular events when we've been studying these drugs for 20 years? And the main issue is that we don't really have great models for studying heart. So we have animals, and there are great things that we can study in those animals, but there are some differences between the hearts in various animals that we study and our hearts. And so you might ask yourself, well, why don't we just take pieces of our heart and study it? And the problem is that our hearts don't regenerate. So if you take out a piece of it, then you've basically given it up, and that's not a good thing. And so you might ask yourself, well then how do we go about creating some model of human heart tissue that we can study? And luckily, there's been advances in the field of what we call cell biology or cell culture. So in most other cells, like in cancers, the way that we study them is that we can culture them outside of your body and then study how they work. In heart, that hasn't been possible because if you take cells out of your heart, like I said, they don't regenerate, and even the ones that you can take out only survive for a few hours. Along the way, what happened was that there's this technology where you can take cells out and culture them that's been around for about 100 years and works for many other cell types. But in some cell types that don't regenerate, it's really hard to culture those cells. And that's true for heart, that's true for brain cells and things like this. There was this discovery about 15 years ago that you could take cells from any part of our body and de-differentiate them into stem cells. And that's called induced pluripotency. So you de-differentiate them into a stem cell that is embryonic-like, and then what people figured out is you can re-differentiate them into another cell type. So you can take a skin cell, turn it into a stem cell, and turn it back into a heart cell. And it's kind of like, you know, the way that people thought about chemistry in the old days, where you could take lead and turn it into gold. And this is something that we just didn't think was possible. So once this technology became available to many of us as scientists, we got really excited because now, for the first time, we could culture and generate cells that we could otherwise never study. So in the context of heart, this has been game-changing. So you can sort of see that the process is that you can take a cell and then you turn it into a pluripotent stem cell, and then you can differentiate it into other cell types. That impact has been not yet clinically realized, so we can't take those cells, turn them into a cell type you want, and inject it back into you, and then all of a sudden you have young cells again. And the reason why is because as we de-differentiate them into stem cells, they gain the ability to proliferate in the same way that a cancer does. And so if you re-inject it back into your body, the worry is that we've now created a cell that might turn into a cancer. The reason why that's really hard to study is because it might take another 20 years after we inject it in you before that cell were to grow into a cancer cell. And so thinking about how you would do a safety trial on people is just unbelievably complex. So the other piece is that when you do take cells and you culture them out of the body, it turns out that those cells don't behave the same way they do as when they are in your body. So you take any cell type, whether it's a cardiomyocyte or other cell, and you take it out of the body and you put it on a dish, piece of plastic, and you grow it, it behaves a little bit like the cell that it used to be when it was in your body, but it changes. It changes its gene expression, which means all of the proteins that are sitting in it have changed composition. The way that it functions has changed, so cardiomyocytes often lose their ability to contract if you take an adult cardiomyocyte and put it out in the dish. It loses that ability very quickly. And so you might ask yourself, why is that? And the reason why is because when it's in your body, it is receiving signals from all of the other cells that are around it, and it is paying attention to all of those signals that are around it. And we believe that how those cells are organized is critically important to how they work. So if you think about like a heart, it's pretty obvious, right? The heart is a chamber. It has to be the shape of a chamber so that when it contracts, it can pump blood. If it was in a different shape, it might not work very well. This is also true for lung and for bone and for blood vessels, etc., right? So the way that the structure is organized impacts how it works. And then the other thing that is quite interesting is that if you look at a particular small piece of tissue, like you scale down to a small piece of heart muscle, what you realize is the way that it is organized, even at this level of a single cell, impacts how it works. So, for example, if you look at the extracellular matrix that is sort of lining every cell, how that extracellular matrix is interacting with the cells that are there impacts how those cells signal and function. If they interact with different types of extracellular matrix, or if that extracellular matrix gets stiffer because of fibrosis of some sort, the cells that are in that tissue change their behavior. They become more inflammatory. If you look at the way that cells talk to each other, what they do is they express proteins that signal across to each other. So in the same way that you might sort of puff perfumes around you and then somebody next to you sort of smells it and they behave differently because of that, cells do the same. So every cell type sitting inside this heart tissue is producing other factors to tell all the other cells that they're there and what their neighbors should be doing. So again, you can imagine that how these cells are arranged in three-dimensional space then impacts who hears each other, who smells each other, and if it was misarranged, then the tissue will behave totally differently. And then, near and dear to my heart, the forces that are experienced inside those tissues impacts how those cells behave. So you can imagine as this tissue is contracting, then certain cells are experiencing forces at their cell-cell junctions, and those forces are then impacting how the cells signal. And if the forces are experienced at cell matrix interfaces, then it changes, again, which receptors are feeling those forces and changes how they signal. And so again, that organization is really important to how those cells work. Now, the thing that I find most challenging over the course of the years of trying to study how this works is that all of these different cues that sort of we classify differently, like cell matrix adhesion or mechanical forces or these soluble factors that are moving around, we think of them as different things, like we can isolate them as separate things. But it turns out the cells don't do that. The cells cause all of the other things to change around them in response to any one of these cues. So if I give these cells a certain soluble factor that makes them happier, almost always those cells change their contractile state, and that contractile state changes how the adhesions function. So you apply forces at cell matrix adhesions, it causes those adhesions to signal differently. And so now, I think in my head, I'm just adding this one factor and I'm studying how this one factor is doing something. But inside the cell, all of these pathways are turning on at the same time. So what that means is it's really hard for us to isolate what causes what in these settings. So to me, the biggest challenge in this space is if we're going to take cells from iPS and generate them and try to get them to function like the way they are in our body, we've got to figure out how to trick those cells to behave like they are in our bodies, right? So we've got to somehow give them all the cues that they would have if they were in your heart and get them to function as if they are in your heart. So we have to understand how these pathways work. We have to understand how to recapitulate it. And you might say to yourself, this is an impossible problem. Well, it kind of is an impossible problem. And so the thing that we've been trying to do is ask, can we somehow break this down and figure out what are the most important cues to give to the cells so that they at least behave somewhat like the way they would in your body and get started. And the way that we've done this as a lab is that we've figured out how to engineer different kinds of environments to put the cells into. So you can think of it like I'm making a bunch of jungle gyms and throwing cells into them and just asking, well, do you care about these forces? Do you care about these junctions? What matters to you? And then we look at how the cells behave and then try to understand what happens. So what I'm going to do today is talk to you about some of the work that we're doing in trying to build little pieces of heart tissue and understand how that works. So the way that people study these iPS-derived cardiomyocytes, inducible pluripotent stem cell-derived cardiomyocytes, is that you take those cells, you make stem cells, you differentiate them into these heart-like cells, and then you put them into different formats. And the most standard format is you put them on a dish and you culture them. When you put them on a dish like this, put them on a dish like this, what happens is the cells are now sitting on this hard plastic, and when the cells try to contract, they can't actually move, right? Because they're kind of being pinned down by the plastic. That makes the cell feel like it's pumping against an infinite resistance, right? So just imagine if I told you you were going to exercise by just grabbing something that was a wall and you pull on it, and this is your exercise, you would feel like your muscles aren't really getting the exercise that it really wants. It actually causes cardiomyocytes to go into an inflammatory state because they like to be able to actually physically contract as they're generating these forces. If you do the opposite and you take these cells and you put them in suspension, so you just lift them off of a dish and let them just float, the cells won't die, they will contract, and so now they're moving, and that's good, except now they're moving against no resistance at all. So it's like contracting in free space. So again, imagine that I tell you to exercise, and what you do is you go into space and you go like this. You're like, yeah, I'm getting a lot of exercise. You're not. And so you actually need some level of resistance. And so what we've been trying to do is find some way to build sort of a three-dimensional heart-like tissue structure that is loaded to an extent that is similar to the way that they would experience loading in your body. So this is a system that we developed. And in this system, what we do is we just fabricate a bunch of wells, and inside those wells we have these two pillars that are sitting up in the wells. And the pillars happen to be made out of elastomeric rubber, so they're soft. And it turns out that if you spin cells down into these wells with some extracellular matrix gel, like fibrin or collagen or matrigel or something like that, then what happens is that the cells, after this gel polymerizes, the cells try to pull on that gel. And all cells do this. If you put them in jello, what they'll do is they'll start to pull on that matrix and try to contract it. That's just what cells like to do because they want to kind of hold on to it. Well, in this case, because the gel is so soft, as they contract, the gel compacts. It becomes much smaller in volume. The water gets kind of extruded out. And normally, they would compact into a little sphere, whatever gel you made. But because we have these two pillars here, the gels get caught on the pillars. You can think of like a basketball net, as that basketball net is kind of shrinking. Now the pillars hook onto this, and all of the matrix kind of gets aligned between these two pillars. And all of the cells are grabbing onto that scaffolding and are now aligning themselves. You can sort of see that in this movie, right? In the end, what happens is the cells are actually quite dense because they've kind of extruded all of the volume of that gel. And they can form cell-cell junctions. So if we do this with iPS-derived cardiomyocytes, then they will compact into this tissue-like structure and actually start becoming mechanically and electrically coupled with each other, becoming a heart-like tissue. So I worked on this as a basic bioengineering scientist. I wasn't necessarily thinking about heart at the time. I was thinking about how do we make different types of aligned tissues like muscle and tendon and that sort of thing. And I was fortunate in giving a talk about this work that there were two cardiac scientists that were sitting in the audience once, and they were John and Cricket Seidman, who are cardiologists at the Brigham. And what they did was they came to me afterwards. They said, we have been collecting patients' cells, blood cells, from kids that have these unusual cardiac myopathies. So places where their heart are somehow not forming good muscle tissue. And they had been studying for 20 years what causes it. They had identified a bunch of genes by sequencing those kids, a bunch of genes that are important in driving the heart tissue to become diseased. But the problem was that they took these cells out, they had been collecting blood, and now they could take the white cells and differentiate them into iPS cells and differentiate them back into cardiomyocytes. But once they did that, they didn't know what to do to study the cell. And so they said, is there any way we could take our cells, put them into your devices, and study what happens? So this was quite exciting for me. I'm a physician scientist, I don't practice medicine, but I'd always wanted to do something that was clinically meaningful, and I thought, wow, what a great opportunity. So we took some of these cells from Cricket and John. Some of them came from patients that had no phenotype at all, so they were a control, and others had this disease that's called dilated cardiomyopathy. And dilated cardiomyopathy, what happens is your heart muscle gets thinner and thinner and weaker over time until it can no longer pump blood effectively, and these kids have to go into transplant. And so what we did was we took those cells that were turned into cardiomyocytes and put them into our devices. And you can just see, this is not one of these things where you have to quantify what's happening. You can just look under the microscope they automatically beat, and you can see that these rubber pillars flex. And just by measuring, because we know the stiffness of our pillars, because we measure that, we can measure the force that those cells generate. And so, because of that, you can see that this patient had a mutation in a protein called titin that is found in the cardiac sarcomere, where they generate force. These guys are not able to generate force very well. And it turns out that in many of these dilated cardiomyopathy mutations that Cricket had identified, they have the ability to contract is somehow altered. So we did this across a few different patients who had different mutations in this same gene. They all had this defect. Now, the thing is when you sequence someone's genome and you say there's a mutation here, there's also many, many other differences between, say, 3 patients, right? And so you can't ever be sure that that gene is the only gene responsible for causing this phenotype. What if we say this, but it turns out there's a second hit or a third gene that is really important for it? So right at that moment that we sort of got this data, there was a technology that was developed that was called CRISPR. And we were really excited about that technology because now you can genome edit cells. And what we did was we took this patient's cells and then generated mutations in this protein. And those are these C lines here. And every line that we generated a mutation in titin, we also saw this defect. So this made us certain that it was this gene that was causing this problem. And so this is something that we were able to do because of our use of this technology. Now, when we went on to study why were those cells that had this mutant titin not able to contract, one of my postdocs took these same iPS cells and wanted to look at the sarcomeres. I told you that cardiomyocytes have these proteins in them where they form these muscle bundles that have myosin and actin across from each other. And those units are formed in boxcar-like structures to create the heart muscle, right? And so when each of those contracts, they sort of pull against each other. He wanted to look at those sarcomeres in these cells. And so what he did was he expressed a protein called alpha-actinin and labeled it with GFP. Alpha-actinin is found in these sarcomeres. And so you can see up here in the upper right is a cell where they're expressing this GFP. And then you can sort of see them trying to contract live. You can see it doesn't move very much because this is sitting on a plastic dish. What he found was that when he suspended these cells from adhesion, that they lost all their sarcomeres. They disappeared. And we thought, that's weird. So he plated them back down on a dish and put it under a microscope. And this is the first movie he got. It was like in the middle of the night. He emailed it to me. He said, you gotta look at this. And this is what he saw. So you can see that this cell is trying to form these sarcomeres, these boxcar-like structures that can generate contraction. And they do it spontaneously. Now what's interesting is that, of course, they don't form these beautiful aligned structures that we see in heart. They're sort of a little bit disorganized. And then the other thing you might notice is that they seem to kind of emanate from specific points. And from there, they're sort of being assembled, right? It turns out that these points, we then went on to study, are places where the cells are forming adhesions to the underlying substrate. So we call these focal adhesions. So there's a specific receptor called an integrin that then binds to the extracellular matrix at these sites. They cluster. And then on the intracellular side, the actin cytoskeleton sort of polymerizes from those points of adhesion. And so what you can see here is that as that actin is kind of being assembled, in this particular case, in cardiomyocytes, they assemble into these boxcar-like structures. You can see in this next movie, this was just to prove that the source of these sarcomeric proteins were anchoring at the adhesions. We labeled a protein called paxillin that is found at these sites of cell matrix adhesion. So then the other thing that my postdoc did is he said, well, what is required for them to kind of build this structure? And what he found was that the cells had to generate force. They had to contract inside their cytoskeleton to cause this sort of assembly of structure between the adhesions. So here's an experiment where what he did was he let the cells attach and just let them form their sarcomeres, and then he added a poison blebistatin, which basically inhibits myosin. It causes myosins to stop generating force. And you can see that as soon as he adds that blebistatin, that the sarcomeres sort of disassemble. So this is really cool because it suggests that sarcomeres have to be assembled in order for cells to generate that beating force, but they need some force in the first place to even form the sarcomeres at all. So it's like there's this feedback loop. And so we've been trying to study how this works, and it's something that we're very excited about. And I want to just give you one more example here. We started studying another mutant, and this is a mutant that is associated with another kind of disease called arithmogenic cardiomyopathy. And arithmogenic cardiomyopathies are ones that are the #1 cause of sudden death in kids. What happens is that everything seems normal, but in fact there's a mutation that's causing your heart's electrical activity to be a little bit abnormal. And at some point, if you're unlucky, it can cause basically this tachycardia and then eventually arrhythmias that will cause sudden death. When people have started studying this, what they found is that most of the genes that are associated with mutations here are genes that are involved in cell-cell adhesion. So where the cells form adhesions between each other. And you might say, okay, that makes sense. If those adhesions are a little bit abnormal, then the conduction of action potentials through those cells may be abnormal. One of the things that has been described in this setting that was not expected is that many of these patients have abnormal contractile function in these hearts. So not only are they having arrhythmias, electrical abnormalities, but they're having mechanical abnormalities. And people have been describing and studying how the electrical abnormalities happen, but they've not been studying mechanical effects. The reason why is because there aren't very many tools to measure the mechanical effects, and we had this tool to do that. So we wanted to study that. So I have a graduate student who then used CRISPR to generate mutations in one of these proteins that's most abundantly mutated in this kind of patient population. It's a protein called aquaphilin 2. I think my title got dropped, but the aquaphilin 2, when you knock it out, it causes this arithmogenic phenotype. Now, you can see here, this is work that she did to confirm that her aquaphilin had a phenotype that was similar to what other people had described, but this is already known, and that is that the action potential within these heart cells is abnormal. It has a longer delay before the calcium comes back up. And if you look at the celljunctions, they're a little bit abnormal in the aquaphilin phenotype versus wild types. But what we were really interested in is there anything mechanically abnormal about these cells? And what she found when she put them into these tissues is that indeed they had this reduced contractility. And so we wanted to try to understand what it was that caused that. And it turns out, after spending some time studying this, she found something that was a little bit unexpected. And that is that she took those cells and instead of studying them in tissues, she studied them by plating them onto soft gels as single cells and then looked at the forces that the single cells were generating to see what was going on. This would let her sort of study those forces in more detail. And when she did, what she found was that these aquaphilin mutants, when they were plated in this format, actually generated more force than normal cells. And this was a puzzle because the cells here were generating less force. So of course, we didn't believe this. We thought this was a weirdo effect. She must have used a different cell line. She had different media. Try it again. She still get this result. We went back and repeated this experiment to see if something had changed in the meantime, and we still got this result. So we just thought, okay, something weird is happening. Well, remember, I told you that the way that cells are structured can impact how they function. So we were thinking, well, maybe the cells here, they're in 3 dimensions, they're connected with each other, there's an extracellular matrix around them, they're generating different kinds of forces than when they're sitting by themselves and sitting on this 2-dimensional surface. And we spent some time trying to think about what's the right way to study this. We ultimately came up with this very simple idea, which actually was Kahan, she did this. She decided to put cells into larger patterns on these 2-dimensional surfaces where the cells can make lots of connections with each other. to ask if the presence of multicellularity was important for this phenotype. And basically you can see here that in the presence of other cells, now the amount of force that they generate is significantly reduced in the mutation. So now it becomes easier to say, well, it wasn't like 3D or extracellular matrix or something about that was different than in our 1D or 2D experiment. It was the fact that you had cells that were connected to each other or cells that were not connected to each other that caused this phenotype to appear. So to study that, she started looking at the cell-cell junctions. Is there something about the cell-cell junctions that does something that causes damage to these cells? And I'm not going to bore you with all the little details. All I'm going to show you is this movie down here. If you look at the normal cells, once they make cell-cell contacts, the cells basically stop moving very much. They're sort of stable. And that's because the junctions are getting more and more stable, the sarcomeres are starting to form, and they're kind of starting to mature into a tissue. But you look at this aquaphilin mutant, and what happens is that the cells, it's like they touch each other, but they aren't satisfied. They keep moving around, and they're moving around past each other. And we think that's because with that junctional protein kind of abnormal, that the cells are not recognizing that they're actually seeing each other. So it's like they're trying to handshake, but one side of the handshake is abnormal. And so they keep trying to do this over and over and over again. So it turns out that when they do that, it causes problems in the sarcomeres. And so I'll show you this. What she did was she labeled the sarcomeres in this purple, and then she labeled the cell-cell junctions in green, again, using proteins that express it so she can record live how they look. And what you can see here is that, again, in the wild type case, the cells are pretty stable. There's some movement, but there's not much going on. But if you look at the PKP2 mutants, what you see is that the cells keep trying to kiss each other, right? When they make this contact, what's happening is that these sarcomeres are not very stable. They keep getting moved around, and you can see some of the sarcomeres starting to disappear. So it's like the actin cytoskeleton that is used to make the cell-cell junction, because it's not making a normal junction, is pulling in more actin. Let's try again, let's try again, let's try again. And because of that, it's causing the sarcomeres to start to fall apart. And that's bad because we need those sarcomeres to generate heart contractile activity. What my point is here in sort of showing this part is to simply say that force, we believe, is a critical organizing principle in organizing the individual sarcomeres. I showed you that earlier with that movie of cells plated down. But even at the multicellular level, forces are organizing how the cells are aligning. I showed you that when we made those microtissues in the first place. The contraction of the cells is pulling the cells into alignment, and then they can form this connected structure. So in the course of that work, we had the good fortune, of being connected with a number of engineers and thinking about this problem at a bigger level. So my lab can only do so much, and we're interested in these very narrow questions of how forces and cells kind of organize. And so we got a chance to put together this grant to pull in many investigators to start asking, can we use this concept of force being an organizing principle to be able to build heart tissue at a larger scale? And this grant called CellMed now involves many different institutions, including several key investigators from your team here, and asking, can we use different kinds of engineering approaches and pull them together in some integrated fashion to build heart tissue for various applications. And part of the NSF's mission, which we embrace very much, is not just thinking about the science component of it, but thinking about the training component of it. How do we bring in trainees at every level to kind of focus on this problem? So what I'm going to do is tell you a couple of things that are going on with other people within this team, just to sort of show you how we can kind of help each other out. So Gordana Novakovic is a professor at Columbia who's a very strong scientist in bioreactor design. And what she did was she took our marker tissue system, and you can see she built a similar system, but in my mind, much more complicated and sophisticated than anything I could do. And in this system, what she did was she developed a system where she could actually electrically stimulate these heart tissues. So like our heart, if you shock it, it contracts, right? And so what she did was she set up the system where she could paste and turn on the contraction of these cells at will. In our system, what happens is the cells sort of contract all at their own pace. So every tissue will contract at different rates. In her case, what she did was that she would then put in these electrodes so that she could turn them on, and when she did, it would trigger that contraction. So she can control how fast every tissue is contracting. And she studied how does that change what happens to the tissues. And what she found was that if she paced them with this, what we call intensity training, where she started just when they formed the tissue to pace them at a very slow rate, and then over days kind of increase that rate faster and faster and faster. What she found is that she could exercise the tissues so they got stronger and more mature than anything that any of us had ever seen before. So we call this intensity training, and in so doing, she was able to cause these tissues to then start to become more and more aligned. The cells started to express different proteins that allowed them to become more and more mature. What I didn't tell you earlier is that iPS cells, when we differentiate them into any cell type, we can differentiate them into something that is like a fetal second trimester, maybe third trimester stage cell. But they're nothing like the cells that we have as adults. They have to keep maturing. And she found that by exercising them this way, it pushed them into this later stage of maturation. So it's not surprising in the sense that as we mature, right, our hearts start to exercise more as we're born and we're moving around, and et cetera. And that actually causes the cells to adjust. Another investigator, Jennifer Lewis, took this microtissue thing and said, can we figure out how to scale the production of a larger patch? Like, it's great that you made these tiny little pieces, but if we ever want to make something that's therapeutic, we've got to make something that is big. And so what she did was she took this set of microtissues, changed the format of the device so that they could make a very high density number of these microtissues in a plate. So she would culture literally billions of cells, throw them into these devices, they would form these little tissues. And then what she did was she suspended the tissues off of those pillars, just washed them, and so they all came up in suspension. And then put them into a 3D printer head, a very wide bore head, and then started printing these little tissues down onto a surface. And it turns out that these cells are still moving around a little bit. You have to do it at the right stage. And by printing them in a certain way, you can actually get these cells to now start to align and connect with each other. So all these microtissues are now reconnecting like clay into an even bigger structure. And in this way, she's thinking about how do you make a full cardiac patch, centimeter scale patch, using our microtissues as kind of a building block. Again, just to sort of show you how it's so important to kind of take your piece and then connect with other experts in other areas to kind of go further. And then the last example I want to give is one where we have been thinking about this aspect of what is limiting about the tool that we've made. We always worry about whether what we've made is good enough for something. And one of the things that I worry about is that these microtissues are sitting on these elastic springs, and so the heart is sort of pulling against an elastic spring. And I know, again, just thinking about exercise as an example, if you exercise by pulling against the spring, right, so it gets harder and harder as you pull, your muscle develops in a certain way. If you lift dead weights, which is a totally different kind of force, your muscle develops in a different way, right? And if you pull against sort of like a dash pot, so it's the same force depends on the velocity that you're pulling at, your muscle develops in yet a third different way. The structure is different, the gene expressions are different, etc. So heart similarly, like our skeletal muscle, develops depending on the type of loading that it experiences. And so if it's loading against this, I've always been worried that we're kind of pushing the cardiomyocytes to form a tissue that is not like the heart tissue that we have. And the reason why is because the way our heart experiences forces is best illustrated in this pressure volume loop. So think of volume like deflection, right? Pressure like force. And what happens is that when your heart is relaxed, it's relaxed and then blood flows into it and stretches the heart, right? So that happens at a very low force or pressure. It gets stretched out. And then the heart turns on. A process we call systole, right? Your action potential turns on. Cells start to contract, but then there's this phase where it increases stress, right? It's pressuring, but there's no contraction yet. And the reason why there's no contraction is that all your valves are closed and the heart is increasing the pressure until the pressure inside the heart exceeds the pressure that's in your aorta. Right? You all have had your blood pressure measured. It might be like 110/70. It has to exceed that 70 in order for the pressure inside the heart to be bigger than the pressure in the aorta, and then all of a sudden the valve opens, right? And now fluid pumps out. And then, as soon as your heart stops contracting, then the pressure drops. Again, at a period where it doesn't change its size, it just relaxes, and then slowly it starts to fill again, right? Well, this force application is totally different from this kind of force application. And so we've been thinking, is there any way that we could try to mimic this? And we realized that we couldn't do it using this spring structure. So this great student, Christos, who is very much part of CellMed, and Alice White, who is an expert in mechanics and 3D printing, and Arvind, you're very Arvind, who's an expert in mechanical characterization, got together and asked, is there something we can do to kind of change this? And we asked, could we build a heart chamber using the same kind of approach that we did before, where we throw cells together in a gel, but instead of having two pillars that would cause them to align in one linear piece of muscle, we made a scaffold that is sort of a cylinder shaped, so that when we threw cells around them, they would kind of form a chamber. Now, if you make that chamber out of something that is extremely stiff, it doesn't do us any good, because now we have this, cells contracting around a Coke can. Nothing happens. So what Alice did was she helped to think about how to 3D print a Coke can-like structure where the scaffolding is made of tiny struts so that this whole structure can kind of compress under contraction. And Arvind took that structure and many, many different designs of structures and started to mechanically characterize them. So he would take his tester and be able to apply force and look at the stress strain curves. So this way we could sort of optimize across many different teams what is the level of stiffness that we want this Coke can to be, and how do we make the structure so that cells will be able to form around it and be able to contract it. And so here is shown just kind of how this works. And I was as amazed as I'm sure you are looking at this. Again, you can see that cells have kind of formed this structure around this spring, and then when they contract, it actually ejects fluid out of the chamber. We can look at how it does it by just putting beads inside the fluid and then putting a microscope somewhere below. Like here, we have it set up inside this microfluidic device, and then we can just put a microscope over here to look at the motion of fluid. Now what you can see here is that this is great. This looks a little bit more like heart, but if you ask what's the loading regimes, it looks symmetric again, because of course, fluid is going in, fluid's coming out, right? It's the same thing in both directions. There's no changes in pressures when this happens. And so the only way you can get differential loading and unloading pressures is you have to put valves in, just like the way our aortic and mitral valve are in our heart. So Christos, with his 3D printing expertise, said, oh, I can make a valve that can stick inside this microfluidic device. So he printed a valve that looks like any ball and check valve. And what you can see is that now if you apply pressures on one side, it closes the valve. You apply pressure on the other side, it opens the valve and fluid can go through. And I'm going to skip that for the sake of time. And then what he did was he has this device where he can put one valve in front of this well, put another valve in front of this well. This valve is pointed so that it allows flow to come in from the well into this microfluidic device and sort of fill in the chamber. And then he has this other valve where he can eject fluid coming out of here. So now you can imagine that as this pump is relaxing and filling, fluid's coming from this well down into this chamber. When this contracts, now fluid comes out this way because this valve closes, right? So you're kind of moving fluid from one well to another. But what's really important here is that you can actually make this well have very low pressure by just putting in only a little bit of fluid. And on this side, you can have a little higher pressure by putting in more fluid there. Why would you do that? That's the way our heart works, right? It's taking fluid from your veins, which is low pressure, generating force, putting it into high pressure, and then filling your aorta. So this is what you need to do to generate that pressure volume thing. So this is what it looks like. So shown here is this channel right here where you'd expect bidirectional flow, and over here are pictures near the valve. You can actually see the valves here, and fluids coming in from one and going out in the other. So if you do a little bit of modeling and look at the velocities and back out the pressures that are experienced within that chamber, what you find is that you can generate a pressure volume. So we're super excited about this because now we can sort of study how does the heart muscle respond to this kind of loading regime compared to what we were doing before. And if in the end it looks like it's the exact same kind of response, then we can say, oh, let's go back to our simpler system. But if it's different, then we can say, oh, we should use our simpler system for certain types of questions and this type of system for other types of questions. I don't have a clock thing, so can someone give me a sense of where we are in time? Okay, perfect. So what I want to do is, I had this whole other section that I'm going to skip, but I don't know how to do that without making you guys see everything. So just close your eyes for a second. But what I'm going to say is, there's a whole other set of work that's going on in trying to not just build the muscle part, but to figure out how to build the blood vessels. And I don't have time to go through that. So close your eyes. I'm going to skip through a whole bunch of stuff that we're doing in trying to figure out how to build blood vessels. And I want to get to, sorry. All I'm going to say there is we're trying to figure out how to build blood vessels. We're not as far along as trying to how we build muscle. And in building blood vessels, we're just getting, as a field, to a stage where we kind of know how to build little capillaries, because the cells, the endothelial cells that line blood vessels, seem to have this ability that if you put them in a gel, they will find each other and crawl around and form these networks. And then they do this magical thing where they can form an open cylinder and then allow fluid to pass through. So we've figured out what are the cues that drive that. And then the other thing is that we can make these little capillary beds, but how do you build bigger vessels that can feed into it? And right now we don't know how to get cells to induce that. So the way that we're trying to figure out how to do that is using 3D printing to, instead of building scaffoldings, building basically empty channels through gels. The way you do that is you build scaffolding that you can dissolve away. So you build scaffolding that then you can pour gels around, you dissolve away those fibers, and then you're left with channels going through those. The resolution of 3D printers right now is such that you can build things that are in the sort of 1/4 millimeter and bigger diameter structures very easily. The smaller ones take too long and are really hard to generate with these kinds of materials that you can dissolve away. So we're going to eventually rely on cells to make the little capillary structures and then 3D printers to build the bigger structures. But it's still something that's very early in stages, and there's many, many more scientists that need to get involved to help us figure that out. But what I wanted to share with you is this idea that there are many challenges ahead in trying to figure out how to build heart tissue, but just think of the heart tissue as just one example of tissues that we need to build. There are bone and arteries and muscle, kidney, liver, many, many different organs where, just like in the heart, the final end stage of failure for many of those organs means transplant. But we believe that if you can make small pieces of these tissues that can replace function in those failing tissues, we can actually prevent the need for transplant. We can use these sort of manufactured tissues for those applications. So we're hoping that some of the technologies that are developed in any one of these areas becomes a technology that can be used in many different areas. And as you can sort of see in my illustration here, I know when I started in my career, I always thought, all the problems are done. It's too late. I got here too late. I can't get involved. And what I can tell you is that these technologies will take decades to really figure out how to make robust, manufacturable, get to people. I was just talking with one of my friends the other day that one of these compounds that they just recently developed that was approved for heart failure was discovered in 1989 and became approved this year. That was after they knew the mechanism, they knew how it worked, they had manufactured the drug, and now they needed to study when to use it. So in this case, we're probably still 10, 20 years from being able to make something that actually could be put in people. And so there's so many problems that need to get solved that I hope some of you get inspired to kind of work in this space. And then the last slide here is just to say that I hope that I've convinced you guys that there are limitations to studying these different kinds of biological problems in animals. And we have this ability to take cells and just sort of study them alone, but that it's really important to think about how do we engineer those cells into structures that are more like they are in your real body. And the process of doing it helps us understand what matters and what doesn't. The fact that we actually can do it has potential for therapeutics from a standpoint of transplant or something like that, but those tissues themselves can also be used for studying disease, right? I gave you examples of studying genetic mutations to help us understand how something works. So I think there are many applications in this space that need to kind of get flushed out. So with that, I can tell you that I rarely walk in the lab these days because I spend most of my time trying to fundraise to get this stuff done. And so all of this is only possible by having a group of really talented graduate students, undergraduates, and postdocs. And so with that, thank you for your attention.
So like I said, as an intro, Chris does a lot of things. Does a lot of things very, very well, and hope you got a sense of that today. So, we really want to thank you very much, Chris, for sharing some of your insights, some of what you've learned over the years, and some of where the community should go with the work that has been done. And on behalf of FIU and Heart Day, I want to give you this token of appreciation. Thank you. So next, I'm going to bring up Dr. Hutchison to talk about the next stages of Heart Day.